Welcome back to the deep dive. We're here to take complex biological engineering, peel back the layers, really, and understand those fundamental systems keeping us alive.
Absolutely.
Today's topic, well, it sounds a bit intense, maybe like science fiction, self-eating. We're doing a deep dive into autophagy. It sounds dramatic, but it might just be one of the most critical cellular maintenance systems in your body.
It's a fantastic concept. And yeah, the term autophagy, it's Greek auto for self, phagy for eating. It sounds pretty hardcore.
It does.
But it's spot on, actually. It's this evolutionarily conserved survival mechanism. Basically, autophagy is a really tightly controlled biological process.
Okay.
The cell takes bulk cytoplasmic material, organelles, proteins, that sort of thing, delivers it to the lysosome.
The cell's recycling center.
Exactly. For very calculated degradation and, importantly, recycling.
So it's not the cell falling apart, it's more like adaptation.
Precisely. Cellular adaptation. When a cell faces serious stress, maybe it's starved of nutrients or just exhausted, it kicks this pathway into gear. It literally starts to, well, cannibalize its non-essential parts, breaks them down.
Into basic building blocks.
Into basic building blocks.
Yeah.
The cell is essentially turning its internal, junk drawer into fuel.
Wow. Yeah.
It recycles those digested contents, generates the metabolites, the energy it needs, allowing it to survive until things get better.
It's like the ultimate survival hack.
It really is. And that's why you see it so prominently in, say, atrophic cells, the ones shrinking because they lack resources.
Right. So our mission today, for you listening, is to go way beyond just that definition. We want to unpack the really detailed molecular machinery. How does this actually work?
Yeah, the nitty-gritty.
And understanding this is critical, isn't it? Because when autophagy goes wrong, if this recycling crew works too much, or maybe not enough, it's suddenly central to a whole host of major diseases.
Absolutely central.
Yeah.
This mechanism, it underpins, well, almost everything from just staying healthy day to day to really significant pathologies.
Like what?
We see autophagy involved in major physiological states, like aging or even intense exercise, as how cells stay fit.
Oh, okay.
But then its failure is directly linked to things like neurodegeneration, cancer, chronic inflammatory diseases. Understanding this complex dance is, well, it's vital now for biology, for medicine.
All right, let's get into the machinery then. If a cell's starting this big self-demolition and reconstruction job, it must be incredibly organized.
Oh, it is. Highly organized.
Walk us through the main steps. How does it start when stress hits?
Okay, think of it like internal cellular construction. The whole project kicks off with step one, nucleation and isolation.
Nucleation. Got it.
So when those stress signals arrive, maybe growth factors disappear or nutrient levels drop way down.
The alarm bells are ringing.
Pretty much, they activate the molecular machinery, and that triggers the formation of this brand new membrane structure. It often starts flat and kind of crescent-shaped.
Okay.
We call this the isolation membrane, or sometimes the phagophore.
And where does the material for this new membrane actually come from? Does the cell just whip it up?
That's a good question, and there's still some research ongoing. But the main thinking, according to our source, is that it's primarily derived from the endoplasmic reticulum, the ER.
Which is that huge membrane network already inside the cell.
Exactly, a vast network. But it's probably not the only source. Other places like the plasma membrane, maybe even mitochondria, might chip in parts for that initial structure.
Why does the source matter so much?
Well, it could influence the type of autophagy happening. If the cell needs to recycle a whole bunch of stuff in bulk, the ER makes sense as a big source.
Right.
But if it's a really specific job, like taking out one single damaged mitochondrion.
Like targeted removal.
Exactly. Then the membrane might actually originate closer to that target. It just points to how tuned this system is. Okay, so once that isolation membrane is nucleated, step two begins, sequestration and formation.
This is where it grabs the stuff to be recycled.
Precisely. The isolation membrane starts to grow, elongate, and curve around the material, the cargo that needs to be eliminated.
What kind of cargo are we talking about?
Could be misfolded proteins, maybe specific bits of the cytosol, or importantly, effete organelles, worn out ones.
Okay.
And here's the critical part. When this membrane completely closes around that cargo, it forms a unique structure, a double membrane vesicle called the autophagosome.
Double membrane. Why is that important?
That double layer is key. It ensures the cargo is totally sealed off, protected from the rest of the cytoplasm before it hits the very acidic enzyme-filled lysosome.
Right, keeps the dangerous stuff contained. It's the delivery vehicle.
Perfect analogy.
So the autophagosome is packed and sealed. What's step 3?
Step 3 is maturation and fusion. The autophagosome, carrying its cargo, now needs to find and merge with the cell's main digestive hub, the lysosome.
The recycling center itself.
Exactly. This fusion is tightly controlled, and when they merge, you get this hybrid structure called an autophagolysosome.
Autophagolysosome, got it.
And once that forms,
Then the real breakdown begins. The recycling engines fire up.
that takes us neatly to step 4, degradation and recycling. Inside that autophagolysosome, you have these powerful lysosomal hydrolases.
Digestive enzymes.
Right. They get activated by the acidic environment inside, and they go to work. They break down the inner membrane of the autophagosome.
Okay.
And critically, all the cargo sealed inside. The resulting products, amino acids, fatty acids, basic metabolites, they're then shipped back out into the cytoplasm.
For the cell to reuse.
Exactly, for reuse.
Yeah.
Enabling survival or maybe growth or repair. It's this incredibly efficient closed-loop system for conserving resources.
That 4-step process sounds incredibly precise, but how does the cell make sure it doesn't just happen randomly? There must be tight control.
Oh, absolutely. Intricate control. It's driven by complex molecular signaling pathways. And, research in just the last decade or so has really shed light on the protein choreography involved.
It sounds complex.
The complexity is pretty staggering, honestly. The whole mechanism, especially building that isolation membrane and the autophagosome itself, hinges on this family of specialized proteins. They're encoded by what we call autophagy-related genes, or ATs.
Okay.
More than a dozen of these ATGs have been found, and their protein products are absolutely essential for managing those membrane dynamics.
So if one is missing.
If you knock out even one key ATG protein, the whole process just grinds to a halt. It stalls.
Wow. Okay, so how does the cell actually switch on this ATG construction crew when it senses, say, nutrient deprivation?
Right. Those environmental cues, lack of growth factors, nutrient scarcity, they trigger a specific group of 4 proteins, an initiation complex.
Like a supervisor.
Exactly like a supervisor.
Yeah.
It effectively gives the green light. It nucleates that initial isolation membrane. It's the point where that stress signal becomes actual structural action.
Now, the part where the membrane grows and seals itself off, elongation and closure, that sounds like serious molecular engineering.
It really is.
Our source mentions 2 specific ubiquitin-like conjugation systems. That sounds technical, but important.
It's where the real precision comes in, absolutely. These systems, they're kind of like the systems cells use for tagging proteins for disposal ubiquitination, but here they're modifying proteins that anchor to the membrane and help it stretch and curve.
Okay, let's break them down. What's the first system do?
All right, the first one involves a protein called ATG12. It gets chemically linked or conjugated to another one, ATG5.
ATG12 joins ATG5.
Right. Then this ATG12-ATG5 pair links up non-covalently, this time with another protein, ATG16L1.
Okay, so you get this 3-part complex.
Exactly. This ATG12-ATG5-ATG16L1 complex, it acts like a crucial scaffold. It directs where the second system needs to work. Without this complex, the cell can't get the right lipids to the growing membrane efficiently.
And that second system, that's the one involving the famous marker LC3.
That's the one, precisely. The second system involves linking a specific lipid, phosphatidylethanolamin, usually just called PE.
PE, much easier.
Definitely. So PE gets covalently attached to the protein microtubule-associated protein light chain 3, or LC3.
Okay, linking a lipid PE to a protein LC3.
And this crucial step depends on that first complex we just talked about, the ATG12, ATG5, ATG16L1 complex. And it requires 2 specific enzymes, ATG7 and ATG3, to make that PE-LC3 linkage happen effectively on the membrane.
So this PE-lipidated LC3, it gets embedded in the membrane and helps it grow and curve.
It's integral to the structure, yes. But its importance goes beyond that, especially for researchers.
Why is it such a big deal in research?
Because this PE-lipidated LC3 stays stuck in both the inner and outer membranes of that newly formed autophagosome.
Okay.
Now, when the autophagosome fuses with the lysosome and the contents get digested, the inner membrane and its LC3 get broken down too.
Right.
But the LC3 on the outer membrane sticks around until that fusion happens. So if you see an increase in the amount of PE-lipidated LC3 in a cell, or if you see these vesicles studded with LC3...
It means autophagy is actively happening.
Exactly. It's the biochemical smoking gun. It's a really powerful, widely used marker to identify cells that are actively undergoing autophagy. It tells you recycling is switched on.
All this complexity really drives home that this isn't just random bulk eating by the cell.
No, not at all.
Scientists used to think maybe it was indiscriminate, right? But now we know it's highly selective.
That's a really critical recent understanding. And that LC3 system we just discussed, it actually helps make it selective.
How does that work?
Well, the lipidated LC3 sitting on the autophagosome membrane can act like a docking station, a receptor, for specific adapter proteins.
Adapter proteins.
Yeah, and these adapter proteins, they can bind to specific cargo the cell wants to eliminate, like maybe one particular damaged mitochondrion or a big clump of misfolded protein.
Ah, so the adapter links the specific trash to the LC3 on the recycling bin membrane?
You got it. It physically connects the target cargo to the growing autophagosome membrane. It ensures the right stuff gets engulfed.
So the cell isn't just, tossing everything into the furnace. It's using these molecular tags to specifically grab the things that need to go first.
Precisely. We even have names for some of these selective processes now, like getting rid of damaged mitochondria specifically is called mitophagy.
Mitophagy makes sense.
Removing worn-out peroxisomes is pexophagy. This targeted approach is absolutely essential. Like you said, the cell isn't just starving and randomly eating its healthy DNA or ribosomes.
Right.
It's prioritizing the removal of damaged components, things that could actually cause long-term harm if they stick around.
Like those damaged mitochondria you mentioned.
Exactly. A great example is how mitophagy often involves another protein called parkin. Parkin specifically tags a damaged mitochondrion, basically putting a recycle me sign on it.
And the autophagy machinery sees that parkin tag and knows to grab it.
Precisely. The autophagy machinery recognizes that tag and engulfs the tagged mitochondrion. It's a perfect example of highly targeted demolition and recycling.
That detail really shifts the perspective. Autophagy isn't just emergency survival, it's the ultimate cellular housekeeper.
It really is.
So, looking beyond just dire straits, what are its constant day-to-day roles in just keeping a cell healthy and well-maintained?
Well, the core function is still that survival strategy. Even during minor dips in nutrients, not full starvation, autophagy helps manage that cellular stress by recycling metabolites. It provides resilience, homeostasis.
Keeps things stable.
Keeps things stable. But beyond just managing resources, that targeted cleaning function is vital.
Preventing cellular decay, basically.
That's the crucial housekeeping job. Organelle turnover. Cells are dynamic. They're constantly making new organelles, but the old or malfunctioning ones need to be cleared out.
Right.
Autophagy is constantly clearing and turning over all the internal structures, mitochondria, even old lysosomes themselves. It ensures everything is functional, efficient.
And we know, especially with mitochondria, if that turnover fails, the consequences are pretty bad.
They're profound. Damaged mitochondria are metabolic disasters waiting to happen. They leak harmful reactive oxygen species, ROS.
The stuff that causes oxidative stress.
Exactly. Leading to inflammation, DNA damage. Autophagy removes them neatly, efficiently, preventing all that downstream damage. If that mitophagy process slows down or fails,
The cell fills up with toxic garbage.
pretty much, it rapidly accumulates toxic junk. And that contributes heavily to many of the problems we associate with aging.
Which brings us neatly to clearing aggregates. That's a huge topic in neurodegeneration research.
Absolutely essential. Clearing intracellular aggregates. These are clumps, basically, of misfolded or damaged proteins.
Like amyloid plaques.
Components related to those, yes, and other types too. They build up naturally with stress, with age, or sometimes due to genetic issues. Autophagy acts like the cell's essential street sweeper.
Keeps the pathways clear.
Maintains cell integrity. If autophagy slows down, like it often does with age, these aggregates just pile up. They literally gum up the cell's internal works. And that leads directly to the kind of cellular dysfunction we see in diseases like Alzheimer's or Parkinson's.
Okay, so we've built this picture of autophagy as survival, resilience, housekeeping. But you mentioned a paradox earlier.
I did.
This system designed to save the cell can sometimes lead to its death. How does that work? How can something protective become destructive?
It's a critical point, and it really highlights the fine line autophagy walks. Fundamentally, yes, it's a defense mechanism.
Right.
But imagine the stressor is just overwhelming. A massive dose of toxin, extreme radiation damage, or just starvation that goes on way too long.
The cell is just too far gone.
The cell's recycling capacity gets completely overwhelmed. The autophagy program starts, it tries, but it's just inadequate to cope with the sheer amount of damage.
So it's like starting a rescue operation that's doomed from the start. It burns through resources trying to fix things, fails, and that triggers death.
That's a good way to put it. The cell essentially passes a point of no return. It initiates autophagy, desperately trying to survive, but the damage is too widespread, too severe. And past that point, the intense autophagic activity itself seems to contribute to triggering cell death.
Is this the same kind of programmed cell death as apoptosis, that clean, controlled suicide?
Crucially, no. Morphologically, mechanistically, it looks different from both necrosis, that messy inflammatory death, and apoptosis.
So it's a distinct death pathway.
It seems to be.
Yeah.
And honestly, we still don't fully grasp the molecular signals that flip that switch, that turn autophagy from a pro-survival signal into a pro-death trigger.
That must make things really confusing for researchers if they see lots of autophagy happening in diseased tissue.
Exactly. Are those cells fighting hard to live, or are they actively dying because of, or at least accompanied by, intense autophagy? It creates this challenging ambiguity.
What do they actually see under the microscope?
Pathologically, you often see the cell filled with autophagic vacuolization, just riddled with these autophagosomes and autophagal lysosomes.
Too many recycling bins, basically.
Way too many, yeah. And this intense vacuolization often accompanies or comes just before the cell's final demise. It's the clearest visual clue we have, but it's still more of a description than an explanation of why the cell finally gave up. It's a major puzzle, a very active area of research.
Okay, let's shift gears now to the clinic, because how this housekeeping system works, or fails to work, is clearly central to so many diseases. Let's start with cancer, where autophagy seems to play this confusing, double-edged role.
Yeah, in cancer, it's genuinely complex. It creates real challenges for developing therapies.
Yeah.
You have to think about autophagy in two phases, really. Prevention and then survival.
Okay, phase 1, prevention. How does it help there?
In the cancer prevention phase, autophagy is generally protective. By cleaning up those damaged mitochondria we talked about, clearing out toxic protein aggregates.
It reduces stress and damage?
Right. It reduces oxidative stress, limits DNA damage, the kind of damage that can kickstart a cell becoming cancerous. It helps keep normal cells healthy and stable.
But then, phase 2, once a tumor actually forms.
That's where it flips. Phase 2, tumor survival. Once a tumor is established and growing, especially deep inside the mass, conditions get tough. Poor blood supply means low oxygen, low nutrients.
So the cancer cells are essentially starving internally.
Exactly. And in that harsh environment, the cancer cells hijack autophagy. They use it as an emergency fuel source.
They turn on their own recycling to survive the stress they're creating.
Precisely. They digest their non-essential bits to recycle nutrients, which allows them to survive that severe stress, resist chemotherapy drugs, and keep growing, maybe even metastasize.
Wow. So if you develop a drug to block autophagy, hoping to starve the tumor,
you risk losing the protective effect that autophagy might have been providing in the patient's healthy cells, potentially making things worse or causing side effects.
it's a real tightrope walk.
Absolutely. The goal is to somehow inhibit autophagy specifically in the tumor cells while leaving it functional everywhere else. Very tricky.
Okay, moving from cancer. The link to the brain seems really strong, especially with neurodegenerative disorders. These diseases are almost defined by junk building up in cells.
The connection there is incredibly strong, yes. And the way autophagy fails can differ. Let's take two big examples. In Alzheimer's disease, the issue often seems to be less about starting autophagy and more about finishing the job.
What do you mean?
It's often described as impaired autophagosome maturation. The cell forms the autophagosome. Okay, it engulfs the waste, but then that vesicle struggles to properly fuse with the lysosome.
So the garbage truck gets loaded but can't make it to the incinerator?
That's a perfect analogy. You get this buildup of autophagic vacuole still full of undigested debris, including stuff related to those amyloid plaques. This failure to clear the waste just makes the disease pathology worse, contributing to neuron death.
And is it different in something like Huntington disease?
Yeah, in Huntington disease, the problem seems to start earlier. The mutant huntingtin protein itself, the one that causes the disease, appears to directly mess with the efficiency of the autophagy machinery itself.
So it's not a fusion problem, it's the whole process being gummed up from the start.
More like that, yes. Yeah. The actual encapsulation and sequestration process seems flawed. But in both cases, Alzheimer's and Huntington's, the end result is similar. The failure of this critical waste disposal pathway leads directly to toxic junk piling up and neurons dying.
It's not just chronic diseases, though. Autophagy has a frontline role in fighting off invaders, right?
Absolutely. It plays a critical role in infectious disease and host defense. Sometimes this specific function is called xenophagy, eating foreign material.
Xenophagy, okay.
When a pathogen, say bacteria or a virus, gets inside a cell, autophagy can be activated to specifically target that invader and deliver to the lysosome for destruction.
So it eats the germs.
Essentially, yes. It's a key defense mechanism against intracellular threats like mycobacteria, which cause TB, Shigella bacteria, even viruses like herpes simplex virus 1.
And this isn't just about killing the bug, is it? It connects to the wider immune response.
It's fundamental to it. That degradation process does two vital things. One, yes, it directly eliminates the pathogen, but two, breaking down the microbe releases its proteins.
Ah, and the cell can then show those pieces to the immune system.
Exactly. Those microbial proteins get processed and delivered to antigen presentation pathways. That's how the adaptive immune system learns what the enemy looks like, so it can mount a specific, targeted attack.
So autophagy is crucial for T-cell activation and things like that.
It feeds into that entire process,
Yeah.
and the source material gives a stark example. If you specifically delete the ATG5 gene, just in macrophages' key immune cells in lab studies,
What happens?
those subjects become much more susceptible to tuberculosis. It clearly shows how necessary that ATG component and thus autophagy is for our innate ability to fight off certain infections.
Okay, one last major connection. The gut. There seems to be a really clear genetic link between faulty autophagy genes and inflammatory bowel diseases, IBD.
This was a huge breakthrough from genetics research, specifically those large-scale studies called GWS genome-wide association
It's
studies.
scanning the whole genome.
They scanned thousands of people and found strong links between both Crohn's disease and ulcerative colitis, the main types of IBD, and specific genetic variations. Tiny differences, single nucleotide polymorphisms or SNPs.
Little typos in the DNA code.
Exactly. And these specific disease-linked SNPs were found right in an autophagy-related gene, ATG16L1.
ATG16L1. So we know genetically a variation in this recycling gene is tied to chronic gut inflammation. But how? What's the mechanism thought to be?
That's the million-dollar question, and where research is really focused now. The genetic link is solid, undeniable, but the function is still being worked out.
Any leading theories?
One major hypothesis involves the role of ATG16L1 in gut immune cells, particularly specialized cells in the intestinal lining, like panic cells. These cells secrete antimicrobial peptides, like defenses.
Okay.
If ATG16L1 is faulty due to that SNP, the thinking is it might impair how these panic cells handle bacteria they get too close, or maybe mess up the secretion of those defensive granules.
Meaning the gut's defenses are weakened, or it can't clean up minor bacterial breaches properly, leading to ongoing inflammation.
That seems to be the direction. Either the flawed autophagy fails to clear invading microbes effectively, or it somehow destabilizes the crucial barrier function of the gut lining. The source itself emphasizes, though, that the exact mechanism connecting these ATG16L1 variants to inflammation is, well, not yet known. It's a major active research area trying to bridge that gap between the gene and the disease.
We have certainly navigated a complex topic today. Let's try and synthesize this for everyone listening. Autophagy, it's so much more than just self-eating.
Way more. It's the cell's incredibly sophisticated, highly regulated, evolutionarily ancient program for survival, for recycling, for quality control.
We've seen it's the difference between a cell just collapsing under stress, messy necrosis, versus elegantly breaking down its own non-essential parts to adapt and survive. That's autophagy.
That's the core of it. Grace under pressure at the cellular level.
So, quick recap of the key takeaways. First, the mechanism. Precise steps involving sequestering material in those double membrane autophagosomes.
Which then have to mature and fuse with lysosomes for the actual breakdown.
Second, it's an adaptive response. Ramped up during nutrient deprivation, letting the cell cannibalize itself to generate fuel and building blocks.
Survival mode.
Absolutely. Third, it's meticulously regulated. That whole construction process needs the coordinated action of over a dozen ATG proteins.
Including those ubiquitin-like systems, especially the one that tags the membrane with PE lipidated LC3, that key marker researchers use.
Critical marker. And finally, when it goes wrong, dysregulation that's central to pathology. We saw its complex role in cancer, both good and bad. Its failure in neurodegenerative diseases like Alzheimer's and Huntington's, leading to toxic buildup.
Its vital role in fighting infections. And that genetic link to IBD via ATG16L1, it's implicated everywhere.
Which brings us to our final provocative thought for you to consider. We know autophagy's efficiency, its ability to selectively clear out the junk, the worn-out organelles, the misfolded proteins, is crucial. It slows down the damage tied to aging and chronic illness.
So the big question is, if we could figure out how to safely, precisely boost that clearance function, enhance that specific targeted recycling, the mitophagy, the clearing of aggregates, maybe manipulating the LC3 system.
Could we fundamentally slow down the accumulation of damage that seems to be at the root of so many major chronic diseases?
Could we move beyond just treating symptoms and actually work towards preserving cellular health, cellular integrity, maybe even extending healthy lifespan, fine-tuning the cell's own housekeeper? That's the potential holy grail researchers are chasing right now. A huge frontier.